Okay, guys, so I'm gonna try and finish off this chapter. It's actually not, it's more of a descriptive chapter. I think the hardest thing is maybe talking about, which I did start last time and we did some progress on, was the polymerase chain reaction. Other than that, it kind of just describes a lot of stuff. It's not all that difficult. It's kind of interesting chapter just on biotechnology and where things are going, which is sort of amazing. So hopefully we'll finish this off today. I don't think it shouldn't be too much trouble. And then your homework is due this weekend on Sunday for this chapter, and then on Tuesday you have the quiz for this chapter. Which chapter is it? It's chapter 26. And then we'll have a review session on Tuesday, and then your final exam will be on Thursday in class, okay? And that's it. So any questions on that? Let me see, in terms of, we have a lab, right? So you do have lab. So next week will be I'll send out also an additional message to my lab people, because I know some of you aren't in my lab, but for those of you that are in my lab, we're having your lab, the final thing will just be a lab practical, right? So you should turn in your final lab report, which was on the last two labs were on this last thing. Did you see the people in my lab? Did you see that I sent you a picture I made of the, well, I posted it on Blackboard, okay, to our lab section of, I used a more powerful UV light source, and then you could see the DNA even better, but you could use your own picture if you want to, either way, okay? All right, so let's get into polymerase chain reaction. So this machine, also just something called PCR, is actually one of the most common machines you find in any biology lab, but it's a fairly recent invention, and the machines just keep getting better and better and so forth. But what they allow you to do is take an incredibly small amount of DNA, and then essentially copy it over and over again. And you can also not just copy the DNA, but copy specific parts of the DNA that you're interested in. So if you're interested in a particular gene, you can make many, many copies of it. Or maybe you have a set of genes that you want to look at and investigate. You could copy those very specifically and then look at those, right? So how does it work? So I did this last time, but I want to try and do a little more. I've got my little cheat sheet here. So we're going to start off with DNA, which is double-stranded. So this is your typical double-stranded DNA. And remember, we talked about how DNA is a fairly stable molecule because of the hydrogen bonds between the two strands. So we have one copy here. I know I'm going to run out of space here. We'll see what happens here. So you want to copy a specific gene. Well, you need to have access to the genetic information within the DNA. So the very first step is what's called denaturation. So I'll put step number 1, and we can refer to this as denaturation. And we've talked about denaturation before in terms of protein and how it can unfold. So what do you do here? We're going to use heat to do the denaturation, and we're going to use 95 degrees Celsius. So that's going to denature our, I can see I'm going to run out of space. Let me get this out of the way. So what happens with the denaturation is you're going to, this will open up this hydrogen bonds, all right? So now this splits and it opens up. So now we have two separate pieces of DNA. So the next step is we want to specifically amplify certain sequences of DNA. So the next step is going to be called annealing. ANNEALING. And what you're going to do is cool things down to about 50 to 60 degrees Celsius. And what you have basically, I should mention that we have a tube. All this is occurring in this little tiny tube. It looks like a little teeny microcentrifuge tube, okay? And but you've added, so we were able to heat up and cool this tiny tube within this machine really effectively because it's relatively small volume. But we've added primers, okay? So let me explain what primers are. So we have primers. These are little snippets of DNA. The primers will anneal to the DNA. Okay, so let's draw what this looks like. So we're going to have our two single-stranded DNA now. Now, what you would have done is, let's say you want to specifically look at one particular gene out of the whole human genome, right? Well, you can look it up and you can get the gene sequence, right? Pretty easily on a computer, using databases and so forth. So what you can do is have complementary, and you just, your companies will do this for you. You'll make what's called a primer. I'm drawing it in green, and it's going to be about 20 bases long, right? Something like that. It's, the textbook, they'll show you like 3. That's not enough. You'll need a fairly unique sequence of base pairs that's only going to occur in that one place, right? And so here's our gene, right? This is our gene that we want. There's a lot more DNA. I'm just simply not showing it to make things simple. Now, the gene is also over here, right? Here's the complementary piece of DNA over here. So we can also design a primer, and it's actually going to start, let's say this is the start of the gene and it goes over here. We can also have a primer for the end of the gene over here, right? Because we're going to make copies of this DNA. So we'll have these will be coding for, essentially we're binding to the same piece of DNA, this little snippet of DNA here. But these primers are different, right? This is going to be a primer that's complementary to the beginning of the gene. This will be complementary to the end of the gene, okay? So, but that's easy enough to look up. Like you say, okay, I want these particular primers. You just go on your computer and you say, okay, send me these primers. And then a week later, you get the primers, right? So it's very easy to synthesize DNA now, which is an amazing statement, but it's very easy now. So now we're going to go into the next step, which is called DNA synthesis. Your book calls it extension, which is a good way of describing this phase. Also, because what we're going to do is you've started with these primers, you actually started the process of replicating. This is the DNA you want, right? So you've added that, you've annealed that. But now, this is only, say, 20 base pairs, right? 20 bases long, right? You want maybe your whole gene is 300, right? So you're going to have to add something that's going to continue this process, right? So then we're going to heat again. We're going to apply heat again, but this time we're only going to go to 72 degrees Celsius. And everything that I'm mentioning, it's all in this one little tube, right? When you start, you have to start everything off in here. So now we're going to use what's called a TAQ or TAC DNA polymerase. Okay. So does the name DNA polymerase make sense to anyone? What do you think a DNA polymerase would do? Yeah, it's an ACE or ACE, depending on how you want to pronounce it. It means it's an enzyme, right? So it's an enzyme. It's going to make a polymer of DNA, right? So what it's going to do, let's go ahead and draw this. So this is our starting point with our primers. Put this in here. You have a bunch of nucleotides, right? So what you've done is, I should say plus nucleotides. Remember, you put out all these things. It's like kind of a soup of things you put all in that little tube to begin with, right? Because you close the machine up and you kind of forget about it. You walk away for 40 minutes, right, while this is running. All right, so what are we going to do here? We're going to extend from you have to have some primer to start you, right? This thing won't work unless you got a little bit of DNA to start. But once you got this thing, this DNA polymerase will come in and they'll work like this. And they'll start taking you have a bunch of nucleotides in your solution, and it starts assembling those nucleotides, making complementary DNA for that strand here. And little by little, it's making complementary DNA for this strand here. Okay, does that make sense? So you're extending out that DNA. And I'm not going to go into details here because it gets a little, it's a little more than we really want to go into, but it actually would continue to go. The DNA actually extends beyond the gene of interest. Okay, but I'm sort of skipping down a little bit because it's just a little confusing. We can come back and address that later if we want to. But we're good here, right? So we've basically made copies now of our DNA. So what's the next phase? What do you guys think? What might be the next phase? Okay. Black this out. We can change the input. Oh, there we go. That's fine. Right. All right. So what's our next step going to be? Can anyone guess? What do we want to do? We've doubled our DNA, right? Because we made copies of it. What might be the next step? We want to make just, if we just double the amount of DNA and we started with a really trace amount of DNA, we haven't really done anything, right? That's still not enough DNA. We want to make literally billions of copies probably of our DNA. So what are we going to do? Do we just stop here? Okay, so one of the key things about these machines is they have little timers in them, and what they do is they cycle around and around and around, right? So so we're going to go right back to this first step here, the denaturation step. Okay, does that make sense? I wish I could. Maybe I need to turn this off. I'll turn this off. All right. So we go back to denaturation. So we go back to that step number one. That's our denaturation step. And we're going to heat everything to 95 degrees Celsius. What's going to happen to our strands of DNA if you heat it to 95 degrees Celsius? They're going to split apart, right? Those hydrogen bonds will get disrupted. So now this becomes two. And this is two strands of our gene, right? So now we got two strands there, right? So then we go to the next step. We do the annealing step, number two. So we're going to cool everything down to 50 to 60 degrees Celsius. And we have a lot of primers. Again, anneal to the DNA. So the word anneal in biology is different than the standard sense of annealing. So annealing in most of the world has to do with when you're working with metal and you heat it up and then it sort of solidifies and so forth. That's what annealing normally is. But in biology, annealing is referring to the sort of primer sticking to the DNA. Okay. So we have our four pieces here. 1, 2, 3, 4. And it's just basically a repeat of what we saw over here. We have the primers come in and they're going to attach here. And then we go on to 3. This is the DNA synthesis phase. And again, your book calls this extension, which is fine. And how are we going to do this? We heat back up to 72 degrees. And we've got our DNA, our TAQ polymerase. And we got a bunch of nucleotides. So remember, we loaded this thing up. Everything we needed, we put it in the very beginning, right? So tons and tons of nucleotides in here. So then we go back here. Here's our 4 strands. And the polymerase does its job, right? It's a big enzyme that comes along and it works its way along the strands. Okay, so how many pieces of DNA, we started here with this one double-stranded, so that's 1 copy. Here we got to 2 copies right here, right? If we're trying to. How many copies do we have now? 1, 2, 3, 4, right? We're at 4 copies. All right. So then you repeat at the end of the third cycle, how many copies are we going to have? This is cycle 1, cycle 2. How many copies should we have at the end of the third cycle? 8 copies, right? Obviously. Fourth cycle, 16. Fifth cycle, 32. Sixth cycle, 64 and 128. 256. 512, is that right? 1024, 2048, right? It keeps going on and on. So within 40 cycles, how many copies are you going to have? I don't know. You have to do the math. Billions, right? It's going to be like in the billions. Every time you double, right? It's incredible, right? Exponential growth of the number of copies. So why is this so useful? Why does almost every biology have one of these things? Again, you can take trace bits of DNA. Like let's say you're interested in human ancestry and you're able to get some DNA out of the bone of a Neanderthal. You can take a little bit of tiny amount of DNA and amplify it tremendously. And then you can look at a specific, also not only you're just amplifying the DNA, but you're amplifying specifically the gene that you're interested in, right? So you can amplify it a tremendous amount of times. At a crime scene, you can find a ridiculously small amount of DNA, right? Humans have lots of DNA. So you have DNA, where are some sources what cells in the human might be left behind at a crime scene that would have DNA in it? Skin cells have DNA, right? Hair follicles, like if you know, the very bottom bit of the hair, there's some cells in there. Most hair is just keratin, so it doesn't have any DNA, but the very base of the hair, you have cells that are alive. Those would definitely have DNA. So little bits of DNA. White blood cells have DNA. Red blood cells, you might think that red blood cells would have DNA, but they don't. Does anyone know why?
When they're born, they eject the DNA.
Yeah. So they don't have a nucleus. So red blood cells don't last very long because of that. But white blood cells do. They have a nucleus. So you can get any blood sample and you'll find plenty of white blood cells. So there's lots and lots of sources of DNA out there. You also can do things like, now they're doing things like tagging things with DNA. Like DNA obviously is a very tiny molecule, right? But you could take any object and tag it just by essentially spraying DNA on it or inside of it or whatever, right? And then the criminal takes it, or the person tries to swap out the genuine article for something that's you haven't tagged, right? And they're like, hey, this is the art thing you gave me. It looks exactly the same, right? But then you could just do a little swab test, do this thing, and you know what sort of tag you added to your thing, right? It's microscopic, no one can see it, right? But if it's not there, then you know that they didn't, this is not the genuine article, that sort of thing. So there's all sorts of interesting ways you can use this technology. All right, so that's that. Any questions on that? Does this kind of make a little bit of sense? This is a very common technique. We have plenty of these. We have a machine like this. We just got here at Hilbert. We got a huge new, even fancier one. Okay, so it's something that we have right here on campus that you'll probably use and get to use here. Okay, so one thing that I just wanted to point out. Let's go back to the presentation. I'll make a few more points and then move on to the next topic. Hopefully that will turn on. Hopefully it's coming up. I should put it. I might have changed the input. All right. Well, let's keep it up and coming back on. Let's so the 3 phases: denaturation, right? We heat to 90, so it's near boiling, 95 degrees centigrade. Then the annealing is 50 to 60, and then the extension is 72 degrees. Okay. So this thing is cycling back and forth a lot with different temperatures. Let me see if I can get the right input now. I messed it up earlier. Oh, there it is. Okay. Yeah, so the basic steps occur repeatedly, usually about 35 to 40 cycles. Let's go on here. So we got the three cycles, the three parts of a cycle: denaturation, annealing, and extension. So you notice that we're getting close to boiling, okay? So the DNA polymerase that we use is not human DNA polymerase, because ours, it's an enzyme, it's made out of proteins. Ours would just essentially denature, right? It's essentially like you're cooking it, right? So what's useful is we essentially got this. There's a lot of different bacteria in the world. This CAC DNA polymerase comes from a type of bacteria that lives in very, very hot conditions, near boiling conditions in a hot spring, right? So it's kind of nice that there's a lot of variation out there in nature, because we can get all these very useful, there's lots of useful organisms that have very useful enzymes for us to use as humans. Yeah, so PCR is a chain reaction because the targeted DNA is repeatedly replicated. The amount of DNA doubles with each cycle. Automation is possible because of the use of a temperature-insensitive DNA polymerase extracted from Thermus aquaticus, a bacteria that lives in hot springs. So literally, these scientists go to places in the United States where hot springs. Where do you find hot springs in the United States? Yellowstone, right? You've probably heard of these people that thought it was a good idea to go swimming at the hot pools, the thermal pools in Yellowstone. It's not a good idea. They're near boiling and they're extremely acidic. So a person will go in and nothing will come out. They'll really find like a little, like maybe like something like plastic might survive, or like a belt buckle or something. That's all we can find of the person afterwards. So don't go swimming there, okay? So it's incredibly acidic, it's incredibly hot, but they're actually living organisms within there, which is amazing. And scientists have been going back there over and over again and finding these really interesting organisms that live there. The enzyme tolerates the high temperature used to separate DNA strands. So because remember, everything is in this one little tube, right? So during the heating phase, all these nucleotides, they all have to survive this extremely hot temperature. Now, this is pretty brief, right? It's only like for a minute or so, right? But it would denature most enzymes. Okay. So this is just a picture from your book. So it goes through all the steps. You can look at this and study it. All right. So let's look at DNA analysis. So some of you are interested in things like forensics. So there's something called the DNA fingerprint, right? And so the way that the term DNA fingerprint doesn't really refer to a specific technique. Multiple techniques may be used for a DNA fingerprint. I'm going to start right now with just telling you what the old-fashioned way was. The old-fashioned way was method, which really isn't used anymore, is to use what are called restriction enzymes. I mentioned restriction enzymes before, right? Does anyone remember what a restriction enzyme does from last time? So restriction enzymes are found in bacteria. They restrict the growth of viruses within the bacterium. How do they do that? That's why they're called restriction enzymes. So bacteria are attacked by viruses all the time, so they have to have their own defenses. So what do viruses have that allow them to replicate within a bacterium? They have their own genetic material, right? So that genetic material is maybe in the form of RNA or maybe in the form of DNA. If it's DNA, it's most likely double-stranded DNA, just like your DNA, just like the DNA of a bacterium. So what these restriction enzymes do is they look for very specific sequences of bases, like it might be GAATC or something like that, right? And then as long as the bacterium doesn't have that sequence in its own genome, right? Because it would end up cutting its own genome and dying, right? As long as we're targeting sequences that are found just in the viruses and not in the bacterium, then the bacteria is golden. Every time a virus attacks it, it chops its DNA up, and then it, the DNA, the virus can't attack it essentially. So that's why we have the term restriction enzyme. It restricts the ability of these viruses to attack and destroy the bacteria. Now, there's many restriction enzymes, like maybe even hundreds, right? Because there's many different types of bacteria. So we, as humans, use those as tools. We say, okay, we want to cut this DNA at this particular location based on this sequence. Let's look it up on a computer, find the appropriate restriction enzyme, just order it from a company, and it'll come to you, a week later, and then you can chop the DNA up. So what they used to do is they take a number of these restriction enzymes, they chop all the human DNA up from, say, a blood sample, And because everyone's individual is different, a little bit different in the DNA, the chopped up pieces might be slightly different sizes between one person and another person. Now, if two people are brothers or something like that, then they're going to have probably very similar DNA fingerprints, right? But okay, so how does this fingerprint look like? So now you've got these chopped up pieces of DNA. Then you run them on a gel electrophoresis, in which you use charge. So DNA is charged, so move an electrical field. Smaller fragments will move faster than the large ones. The large ones get left behind. And then you get a very distinct banding pattern. That's the DNA fingerprint for that person. You compare that to the DNA fingerprint of the different people that might have committed the crime. And then you look at the blood at the scene of the crime and do a comparison. And again, from blood, you can't get it from red blood cells. It has to be from white blood cells. So that's the old-fashioned way. We really don't use that anymore. There's a newer, better method, and it relies on something called short tandem repeats, or STRs. This is used in essentially doing the same thing as genetic fingerprints. So what is this? Well, short tandem repeats are short sequences of DNA bases that reoccur several times. So if you look at this sequence here, you have GATA, right? But then you notice it's GATA again, and then GATA, okay? So you have lots of these weird bits. You have literally thousands and thousands of these STRs within your genome, okay? And how many repeats? It could be from 5 typically to 50. So in that range where you're repeating, so this is just a very short one here because we've done it 3 times, but it could typically be more like 5 or maybe even 50. And in some cases, it could even be hundreds, but usually that's a little unusual. That's, I think I might have mentioned Huntington's disease early in this course. So sometimes if you have these short tandem repeats and they become out of control, where now instead of more than 50, now 100 or 200, then often that leads to sort of disease states like Huntington's disease. Okay, so we have these weird bits of DNA. They don't code for anything. They're non-coding bits of DNA. So because of that, well, let's think about it for a second. If you have a mutation in an enzyme, in a bit of DNA, it's a gene that codes for an enzyme that's required for glycolysis. Are you likely to are you likely to be living if you have a problem with the gene involved? Yeah, so you die right away, right? So if you have a mutation, there's a good chance. I mean, if it destroys the enzyme, if it's a significant mutation that causes that enzyme to be destroyed, and there's no way around it in terms of your cellular machinery, and it's involved in, say, glycolysis, the cell would just die. It just can't live, right? But if you're in a non-coding part of your DNA, and you have one person has 5 of these repeats, another person has 10, it really has no significance whatsoever on your survivorship, right? You're gonna be fine. You're gonna be able to pass your genes on to the next generation. Everything's fine. So because of that, when mutations arise, which means different numbers of these repeats, typically the whole repeat will be just copied over again, right? So you'll get lots and lots of what's called polymorphism, meaning that across humans, you'll see lots and lots of variation in the number of the repeats within a region of short tandem repeat. So again, people within the same family might have very similar numbers, but people that are from distant families would be very, very different. So they specifically look, so again, we have thousands of these regions, right? But just through experience, they've picked regions that are very polymorphic, meaning that people vary a lot in these particular regions. In the US, we look at 13 regions, 13 STRs across the human genome. In UK, they look at 17. There's really not much difference there. That's a lot of sites, right? So if you then do the statistics with that, and you look at the differences between different people for that many sites, it's very unlikely that you're going to get a match. Almost everyone's very different, except again for like a close sibling or something like that, like a sibling or something like that. So fragments and samples are amplified by PCR are different lengths because each person has his or her own number of repeats at the particular location of the STR on the chromosome. The more STR loci, remember loci is plural locus, employed, locus is a site on a gene, on a chromosome, the more confident scientists can be of distinctive results of each person. Again, this has been around for a long time. People know how to do this statistically and so forth. And again, they look at many sites. So, STR profiling, PCR is used to amplify fluorescently labeled target sequences of DNA. So now we have, so what you can do is, again, we can even do this here at Hilbert. We add something into this here, this initial thing, that's essentially a fluorescent dye, and it'll label the DNA. So you'll get readouts of the lengths of the DNA you have. So laser then detects and records lengths of DNA fragments. And there's different methods here. One is to look at the lengths. Another is just to look at the amount of fluorescence. Okay. The greater the number of STRs at a locus, the longer the DNA fragment will be. Oh yeah. Okay. So we're looking specific to length. So I suppose you would use the laser here. The greater the number of STRs at a locus, the longer the fragment will be. So the more repeats you have, the longer this little piece of this fragment of DNA will be. That makes sense. Can be used to identify samples at a crime scene, detect genetic disorders. Oh yeah, if you're looking for genetic disorders in a person, like there's a whole huge business on genetic counseling, which is a good career, by the way. Like you can have people go and have genetic tests to see if they have a particular gene that might affect their offspring. And then people need to interpret that. So there's a lot of people employed as counselors to help people understand what's going on with their genes. And their relative risks and so forth. And then identify relatives. So you may have seen situations where there was just recently in the news a woman who was abducted 50 years ago, and now she's being reunited with her parents. You see that in the news? Anyway, how do they identify the relatives? For sure, well, they do the DNA sequencing, right? All right. So what does this look like? Here, obviously, we're not showing you all 13 short tandem repeats, but we're just looking at 2 here. So here's just because otherwise it would be very messy and hard to interpret, but we're looking at 2 different sites, 2 different locuses, so 2 loci. At the crime scene, we find for this one particular short tandem repeat locus in the human genome, 12 repeats. For another locus, we find 16 repeats, okay? These are just markers to give us over here in this column, they're just telling us how many repeats they have. Here's the crime scene, we can read it off. We've got 12 and 16, okay? So this is just like a calibration bar essentially over here. Now we've got the 2 suspects here. This guy's got, for locus, the first locus, 12 repeats, and the second locus here, 12 repeats again. This person has 12 repeats for the first locus, 16 repeats at the second locus. So who's the more likely to have committed the crime, suspect A or B? What do you guys think? Yeah, B, because if you look at this, suspect A, for this particular locus, he's only got 12 repeats, whereas at the crime scene, again, using white blood cells from the blood, they've got 16. This guy, he's got 12 and 16. Now, this is not enough evidence. Just look at 2 locuses. There's enough you may not get the correct thing. That's why we look at 13 in the United States and the UK 17. But once you look at many locuses, if you do the statistics, it becomes very unlikely that the person would show up and have matched the number of short tandem repeats at certain loci. So instead of just 2 sites, we look at 17, and it becomes almost impossible to say, oh, this person wasn't at the crime scene. Because there'd only be a couple people in all of the UK, for example, or United Kingdom, that would actually have that profile. Go ahead. Do you have a question?
My question was like, how can you tell? Like, I thought it was A, but how does it be?
Oh, because look, this person at the crime scene, the person was maybe caught like by some glass escaping through the window or something, like they stole a Van Gogh painting or something, right? So they cut themselves on the way out, so they left some blood or white blood cells, which have nuclei, you can put DNA out. And then this person, he had his DNA would just line up here. So both STRs would end up here too. So you have sort of a double bar here. But this person actually has DNA that matches that at the crime scene.
Okay, I thought it was there were 2 PCR tests. I thought they were comparing each one.
No, so we're just showing 2 because it'd be hard to show 13. So each one is a locus, right, on the DNA.
Yeah,
This is
I
the
thought
longest one, this is 2, and of course, we do a whole, all the way up to 13 different loci. And it'd be a very complicated
otherwise
diagram.
like 12 would be compared to the 16.
No. You're comparing between this, right, so 12 and 16. So here are these, all 3 of them have the same number of repeats for STR1, okay? So this is a location, right? But if you look at the second locus, right, on the chromosomes human genome, this one has 16, this one has 12, so it seems unlikely this person was there, right? But this person has 16, so at least it's possible. Now, a lot of people are gonna have 16, right? So if you have a whole country, like a lot of people have 16 there, but then if you look at another site and then it matches again, another site, it becomes almost statistically impossible. Okay, that it wasn't that person. I mean, there might be a couple other people in the whole country that would have that same. And if they happen to be in a completely different area, you can sort of rule them out. All right, so I think that's the hardest bit to understand of this whole chapter is understanding first the PCR and then this historic tandem repeat. The rest of it is kind of fairly descriptive. So we're going to go through this fairly rapidly. So I talked about transgenics. So transgenics means you move something across from one species to another, right? So right now there's lots of transgenic bacteria. There are plants, animals that are often called genetic. And these transgenic things are, by definition, they are genetically modified organisms because you've changed their genome. You've moved a genome from one organism to another. And the products, the products produced here are simply referred to as biotechnology products. So how do we do this? And what's it useful for? Well, transgenic bacteria have been used for many years now. They're produced by using recombinant DNA technology. So I know I'm throwing a lot of words out here, but transgenic means there's just genes from 2 different sources across different species. Recombinant means you're combining DNA. So the term recombinant means it could be DNA. It doesn't have to be transgenic. It could be from you're just combining DNA from two different sources, right? That's all that means. So not necessarily cross species. But transgenic is it has to be recombinant because you're combining DNA. The DNA is grown in these gigantic vats. So they have these factories with absolutely gigantic vats with everything's perfect for the bacteria to grow. The bacteria will then express the cloned gene. We talked about cloning last time. So gene product is collected from the media. So a good example might be insulin is one of the first ones that was used here. So biotechnology products produced by transgenic bacteria include insulin. So we used to get insulin by, so insulin is produced by the pancreas, right? It's a hormone and it helps with regulation of blood sugar, specifically glucose. So it's a fatal disease essentially, but what they figured out many years ago was how to get insulin from cows, okay? And they actually, in the very early days, they did dogs and things like that also, but it sort of settled in on using cows, so bovine insulin. Now, bovine insulin is slightly different than human insulin. So they wanted to give human insulin to people, and they said, okay, well, we can take the human gene for insulin and put it into bacteria and then grow it. And that's how we now have these different companies that generate insulin, okay? Now, some people are missing human growth hormones. It means they'd be very short as adults, but we can overcome that nowadays because we can do the same thing. We can introduce the gene for human growth hormone, which will make the peptide. The gene makes the peptide, so now we can make lots and lots of the peptide and then give that to people as go ahead.
So would that be like injections into their spine?
Yeah, you would just inject it. You just take insulin shots every once in a while. Yeah, I mean insulin shots or human growth hormone shots. Yeah, you wouldn't need it continuously, probably once a week or so. I don't know. I'm not really sure. I'm not an expert on that, but you could just have it injected into your blood. It's a hormone, so it normally circulates in your blood.
Oh, I would just think because of the being a hormone to extension growth, it would be spinal cord.
No, but the point with injecting the blood is it essentially goes everywhere, right? Right, yeah. Another one that's really helpful is if you have a stroke or a heart attack, they determine that it's because of a blood clot, right? So blood clot may have gone and blocked the coronary artery, or blood clot may have traveled from heart of your body to your brain and blocked the blood vessel. That's called an embolism when you have a blockage. But if they can figure out that that's the source of the problem, that you have this blood clot, and again, they'll do scans and stuff before they inject you with anything at the hospital. If they determine, well, it's a clot that's the problem, then they'll inject you with the tissue plasmagen activator. This is a protein that breaks down blood clots. So it's a very helpful protein. It's very helpful medicine, so it can save your life, right? Again, so if someone is having a stroke, or you suspect a stroke or heart attack, you got to get them to the hospital immediately so they can start doing tests and figure out if they should inject this or not. Hepatitis B vaccine. So lots and lots of really useful products, okay? That help people, right? Other uses of transgenic bacteria that live on plants have been altered. So if you think about strawberries, you can only grow them usually in the late, when we get strawberries, sort of in late summer, right? And by the time fall comes around, strawberries get much more expensive. They can be grown in greenhouses, but the ones that you grow outside kind of disappear, and that's because of frost, right? So it gets very cold at night, frost appears in the strawberries that destroys them. Now they figured out that there are bacteria that live on the strawberries, and that actually essentially promotes the frost, right? So what they've done is develop other kinds of bacteria. They're called frost minus bacteria, and then they sort of infect the strawberries with those. That sort of crowd out the frost plus ones, and that makes the strawberries frost resistant. So you now can have frost later into the fall. Okay, so that's helpful. Bacteria that colonize. Oh, another possibility, we have bacteria already that colonize the roots of corn plants. So what they did was they said, okay, the bacteria are already there. Let's engineer them to have genes that are toxic to insects to damage the roots. So that's another possibility there. There are other sorts of bacteria that are very helpful. They can be selected for the ability to degrade a particular substance. So this can be enhanced by bioengineering. So you grow bacteria that can, for example, eat oil. So there was a big, Exxon Valdez? No, it was the Deepwater Horizon in the Gulf of Mexico spilled tons and tons of oil, and it was a big ecological disaster. It was around 2010 or something like 13 years ago. So potentially you could use these bacteria to then eat up the oil, convert it into harmless products. Coal, so really good quality coal doesn't have a tremendous amount of sulfur, so it's less horrible to the environment, but poor quality coal, which are now starting to rot. Of course, we use the good stuff up first, right? So now we're stuck with the not so good coal. It has a lot of sulfur in it, so there are actually bacteria that can remove the sulfur from the coal. So that's another helpful use of biotechnology. What about plants? So let's go on to transgenic plants. So foreign genes can be introduced into plants. There's a couple ways to do this. You can do, and you essentially introduce the genes into immature plant embryos. Another thing you can do is take a plant cell and create what's called a protoplast. You can remove the cell wall. We talked about what it's made out of, mostly cellulose. So you dissolve away the cell wall. You've got a living cell still called a protoplast. And then you can expose this protoplast to electrical current and the liquid around it. So you have a little chamber. You have the protoplast in there. There's little cells that you've removed the plant cells without cell walls. You apply this current, high voltage current. This creates these tiny little pores, these tiny little holes in the membrane, but without destroying the cell or killing it. Now, in this water solution around the protoplast, you have the DNA that you want to enter into the plant. The DNA enters, you stop the current, and then this pores seal up very quickly, right? So your membranes also will seal up very quickly. Pores they're not supposed to have pores. After the DNA is in there, so the DNA is now sort of locked inside. And then you go on and develop these, and they'll generate their cell walls again. And often these protoplasts can make new plants, right? And so now you have these baby plants that have a new gene incorporated into them. So there's different uses for this. One is the pomato. Now you can do a pomato by just grafting together the roots of a tomato with the stem of the roots of a potato with the stem of a tomato. Now you get tomatoes and potatoes right from the plant. But they've also done this with technology I just mentioned here, where you make a protoplast and then you introduce the genes to sort of combine the two. Okay. So things can be done in the old way, fashioned way, or they can be done in the newer way. You can introduce pest resistance into cotton, corn, potato strains. Okay, so you want to make them resistant to locusts or whatever it is. This is the next one's used a lot nowadays. So we have, for example, soybeans that are resistant to herbicide. We also have corn that's resistant to herbicide, that sort of thing. The unfortunate thing about this is this means that if they're resistant, now they can put a lot more of these chemicals. So, yeah, I don't know. There's good and bad with these things, these technology, right? So plants can also be engineered to produce human protein. So that's an interesting one. So you can actually, instead of using bacteria in those fancy bioreactors, you can actually get plants to produce human proteins that are really helpful. The proteins will end up in, say, the seeds of the plant, like maybe it's human growth hormone or something like that, and then you can make your medicine from that. Okay, so transgenic animals. Technology has been developed to insert genes into eggs of animals. It's possible to microinject foreign eggs by hand or by vorticizing. Now, when they say by hand, what are they talking about? You're not actually doing it just with your hand, right? What they mean is, here's your cell, and then you bring in what's called a micropipette, but it's been pulled, it's glass, and you can pull glass through. It's property of glass. You can apply heat and the fancy machine that then pulls on the glass, and you get an incredibly fine tip. And you can then inject your DNA directly into the embryonic cell. Okay, that's what they mean by hand. Hand with the assistance of really high-tech stuff, right? Or by vortexing. This is kind of an interesting method because what they do is they take these little eggs for animal eggs, and then so we're just talking about a single cell, perhaps it's unfertilized egg or something like that. Or maybe it's a zygote, like, you know, it's not clear if it's fertilized or not, but you're at that single stage level anyway. You agitate it with this DNA with the DNA that you want. You want to add the DNA in, and silicon carbide needles. The needles poke little holes, and then allow the DNA to enter. So that's kind of an interesting way of doing it. When the eggs are fertilized, the transgenic offspring are produced. The gene for bovine growth hormone has been injected. So you can get transgenic animals this way. So bovine, of course, is for cows, right? So this bovine growth hormone, that gene can be added to fish. That means your fish now grow much faster and larger. They've done this to things like salmon. Have you seen this before? They have farmed salmon that grow really, really quickly. Cows, pigs, rabbits, and sheep. So we can get all these animals to grow much faster if they have this bovine growth hormone. Gene farming. So transgenic farm animals can be used to produce pharmaceuticals. So this is another really interesting use of transgenic animals. So you can have genes that code for therapeutic and diagnostic proteins are incorporated into the animal's DNA. So proteins are then harvested in the animal's milk. So plans exist to produce drugs for treatment of cystic fibrosis, cancer, and blood diseases. So how does this work? Well, basically, I think there's a picture. Let's see if we can find a picture here. There it is. Production of transgenic animals. So here's a nice picture. We have the human gene for growth hormone. So again, there's a number of people out there that just simply don't produce enough growth hormone. So they would be very short in stature if they didn't get this injection of growth hormone. So we need to produce growth hormone. So we inject it in we micro-inject the human gene. Again, not with a syringe, as they're showing here, but it's a very, very fine glass needle. It's now the gene is now in here, right? So the zygote develops, and you you do essentially in vitro fertilization. This then goes into a host animal, right? So this is a surrogate mother. It makes the baby goat comes out, and now this baby goat can make milk that has human growth hormone in it, and you can extract the human growth hormone out and then inject that into humans. So there we go. So that's very helpful. So that's one example, but we're thinking about trying to treat cystic fibrosis and cancer and blood diseases that way. By blood diseases, mostly we're talking about things like hemophilia, right? People that are missing clotting proteins. So this would be an easy way to make clotting proteins that could help them. Another really interesting technology that scientists use is if they want to understand a gene, they can ask the question, well, what happens if we don't have this gene at all? We can just remove it, right, from the organism. And that's called a knockout, just like in boxing, you know what a knockout is, right? So they're literally just knocking the gene right out of the genome. So this was mostly done with mice, but I think they're maybe extending this to other organisms now. But knockout mice have both alleles for a gene removed, and therefore it doesn't create the protein. It can't create that protein, right? So then you can look at the phenotype without a gene to be studied. Why is this helpful? Well, for humans, we know that there are some diseases where we're missing particular genes for particular products, right? And so this is tragic because then the people often don't live very long, or perhaps they die in early adulthood, right after perhaps having a family and so forth. Huntington's disease is one I mentioned earlier, but there's a whole slew of different genetic disorders that are really tragic. And the problem is, you know, if you're a doctor or a scientist, you're trying to help these people maybe live productive lives, despite the fact that they're missing a fairly important gene, is coming up with diets or regimes and all sorts of things for that is difficult because you have a limited number of people. These are often fairly rare diseases, right? So what you really want is many, many experimental subjects to test with different diets, different medicines, different, you know, exercise regimes, whatever it takes, right, to get the person living longer. How can you do that? Well, actually, you can make mice that essentially have the disease because they're missing the gene also. And now you have hundreds of subjects, right? You can say, okay, well, what happens if I alter the diet in this way? What if I try this particular therapeutic drug? What if I try this other therapeutic drug? What if some combination of diet and exercise? You know, all these things, you can try all these different combinations that you absolutely cannot do with humans, right? So now this allows us to use animals to essentially test out ways to help people, okay? All right, here's just a nice illustration. This, of course, it's mislabeled. It says transgenic animal. Everyone knows this is not an animal, right? This is an apple. These are apple slices. So on the top, you have apple slices that are brown, right? So that happens within a few minutes once you slice. But these ones have been altered so they don't brown as quickly. So that's another example. Okay, we already talked about this. Oh, this was just a continuation. You see, once you have this, once you get your goat, is this a goat? That's a goat, right? That's a goat that's producing milk that has the human growth hormone. Maybe you want to keep that around, right? So what you do is you take cells from this cell, this organism here, that's making your special milk, and then you put that into enucleated eggs. So these eggs have their nucleus removed. And then you put the genetic material from this organism that you want to keep. Like maybe it took you 10 years maybe this process took you 10 years to get that one goat, right? So you want to keep that going. So you take the cells from that goat, put into these nucleus where you remove the genetic material. So these are eggs, right? Those eggs then develop in the surrogate mothers, the host goat, and then you get essentially clones. These are perfect clones of the one that we had earlier, and they're going to continue to make your milk with the human growth hormone. So let's look at gene therapy. This is an insertion of genetic material into human cells for the treatment of genetic disorders and other illnesses such as cardiovascular disease and cancer. So the various methods of gene transfer that can be used. There are viruses. So I don't know if you know this, but a huge percentage of your genome is actually viral DNA. That's because there are certain viruses that actually work by incorporating themselves into your DNA, and they'll actually hide out there for a while, and then occasionally they'll sort of become activated, and then they'll generate more essentially baby viruses using the cell machinery of your cells, and they'll burst out killing your cells. So there are tons of these viruses that have incorporated over the years their genetic material into your genetic material, right? Into your ancestors' genetic material. So we can take advantage of that because we have viruses that can be genetically modified to be safe, and they can introduce normal genes into your cells, right? Into the nucleus of your cell. Another way of doing this is liposomes. These are microscopic globules of lipid. I think I might have briefly talked about liposomes before. Does anyone remember what they are? No. That's lysosome. It sounds similar to lysosomes, right? Liposomes are different. They look like this. So remember when we talked about, so we have one thing called the micelle. So if we look at, we talked about phospholipid bilayers. So one thing that can be formed is if you make some artificial phospholipids, they may form into something like a bilayer. They could form micelles, looks like a little droplet. You see all the hydrophobic tails are pointed away from the water, and the polar heads are pointing out, right? That's a micelle, but now a liposome looks like this. Maybe I didn't talk about this. I'm gonna get tired of drawing, but so it's going to look like this. I'm going to get tired of drawing this, but you see what you do is you continue this all the way around until you have a sphere, right? So this, in the middle of this is where you put your drugs, your therapeutic drugs in here. Now, why is this useful? Well, because you can inject this into the person. This is like a lot like a phospholipid bilayer. It's artificial, right? But this thing, once it comes in contact with the phospholipid bilayer of one of your cells, will actually fuse. And the drug, which is outside of your cell, now ends up inside of your cell. Okay, that's a liposome. All right, so they're microscopic globules of lipids, but they have fluid in the middle, and you have the drug in the middle there. They can be used to introduce normal genes. So in this case, instead of drug, you put genes in there, right? Sometimes the gene is injected directly into a specific region of the body. You can also do injection. All right, and here's just some examples. Why do we care about this stuff? Why is it helpful? Huntington's disease is a terrible disease of the brain. Alzheimer's disease, a huge percentage of people will get Alzheimer's disease. I forget the exact percentage, but as you get older, the chances get very high. Parkinson's disease is a very common disease for older people. Brain tumors occur pretty frequently also. So there's a potential therapies for all those things. Skin cancer is a big deal. So we can try and modify genes there. Lungs, cystic fibrosis is really tragic. People don't live very long with cystic fibrosis, although the survival rate is getting better and better with new technology. Hereditary emphysema is bad. Liver, we have familial hypercholesterolemia. This is where you have high levels of cholesterol in the blood because the liver cells do not have the receptor that allows them to pick up the cholesterol and then modify it and so forth. Blood, sickle cell bone disease is pretty horrible. So perhaps we can help people by doing a bone marrow transplant. Endothelium, this is the blood vessel linings. These can be transferred by implantation. So hemophilia, when people missing clotting proteins. Diabetes mellitus, that's another possibility. Muscles, muscular dystrophy, you've probably heard of, right? This is where people get very weak and eventually they die. It affects mostly boys and not girls, but it's the most common one is Duchenne muscular dystrophy. It's really tragic disease, but now perhaps we can inject genes directly into muscles, which are affected in this disease, help them live a little bit longer, hopefully better. Bone marrow, we can transfer things. So again, sickle cell anemia is a possible thing there. So let's take a look at something. So ex vivo gene therapy. So what is ex vivo gene therapy? This means you take a sample from your patient, remove it, modify the genes within that sample, and then inject the sample back in. So vivo means life. Ex means, like, you can think of what the word exit, like, so it's outside of life, right? So you take a piece of the liver, for example, you take a sample of liver from a person, you remove it, modify the liver cells, it takes a week or something, put them back by surgery and the person, that's ex vivo. You've taken it out and you put it back in. So here's one method for ex vivo method for treating severe combined immunodeficiency. These are kids that essentially can't ward off disease very well. So again, very tragic. They can die very young. This is used for children who lack the enzyme ADA, adenosine deaminase, which is involved in the maturation of two very important types of cells. T cells and B cells are very important immune cells, part of your immune system. They're prone to constant infection. They may die without any treatment. Okay, so what can we do to help these poor children? Gene therapy treatments steps involve this: removal of bone marrow stem cells from the body. Infected, and so now you have essentially the bone marrow cells in a dish, right? That's outside of the body, a glass dish. You infect the cells with a virus that carries the normal gene that codes for the enzyme ADA, adenosine deaminase. So this will help with the maturation of these immune cells. Return the cells to the patient with the hope they'll divide and express the normal gene for ADA. So now they may be able to produce the product that's needed for the maturation of these cells. Okay. Let's go on. Here's another one. Another one really tragic one is familial hypocholesterolemia. I think I showed a picture of this earlier in this. Did I? I can't remember if I did. I taught so many classes. But basically, if you have cholesterol, these people have essentially no ability to remove the cholesterol from the blood, so it gets built up to very high levels. This is really horrible, right? It can lead to things like heart attacks. High cholesterol levels may make a patient subject to heart attacks at a very young age. So liver cells, what happens in them is the liver cells lack a receptor protein for removing cholesterol from the blood. So what do? They take a portion of that person's liver out, obviously by surgery. They infect that, again, now it's in a dish and it's under sterile conditions. They infect this portion of the liver with a virus containing the normal gene for the receptor. They put this portion of liver back into the patient. Obviously, it's very difficult, right? They have to hook up blood vessels and all sorts of things like that. And then hopefully they'll survive. And then now they'll be able to start removing some of the cholesterol from their blood. Oh yeah, this is the picture. This is the one we're showing this. Yeah, we're looking. This is the one I think for the children, right? So we remove some bone marrow from the children. We have the defective gene. Use a retrovirus to bring the normal gene into the bone marrow cell. So retroviruses are going to be viruses that are able to take their DNA and incorporate it into our genome. Okay, so that's actually fairly common. I mean, a lot of viruses do that. I'm looking at my notes here. Yeah, anyway, that's what those do. Okay, so then so this is the normal gene. It gets incorporated into the DNA. You can see here the picture of the normal gene is now. Well, it may not go to the right place, right? It's just been tacked on to the chromosome here. Then you return these genetically engineered cells to the patient, and hopefully they'll divide and do fine. And now they can generate this the protein product that they were missing before. Okay. So there's also in vivo gene therapy. In vivo means in life. So you don't. So basically, we're going to inject the gene into the person in various ways, and then we hope that the gene sort of sticks. So here's one typical method here. Cystic fibrosis patients, they lack a gene for coding for chloride transporter membrane protein. So why is chloride important? If you look in the lungs of these people, you have let's look. This would be the case for your lungs. So you have cells. So this is, let's imagine that this is a passageway, a small passageway within your lungs. So there's air and there's a little bit of water and fluid along the edges here. So you're going to have some cells, and they normally would transport chloride, which is an ion. They're also going to transport sodium. Sodium chloride, what is sodium chloride? Does anyone know? Salt, right? That's table salt. So you're going to have a bunch of these cells. They're moving chloride and sodium over this way. Now, if you make kind of a salty solution over here compared to over here, what's going to happen? Which way will water move? Does it move into a salty solution from a less salty solution? Which way would it move? Yeah, so water will always move from an area of high water concentration, right, to an area of low water concentration. So here the sodium chloride are taking up space, and so the water is going to move also, right? So the water essentially follows the movement of these ions. Whoops. So why is that good? Well, it's because you want mucus to be actually fairly fluid. Why does it need to be fluid? Because you have cilia within the lungs, and they're pushing the mucus up all the time, from very small passages to bigger passage, all the way to the windpipe, right, the trachea. And then what happens to that mucus? You end up swallowing it just sort of subconsciously, right? And that kills all the bacteria. It gets rid of all this horrible pollen or whatever you inhale, right? So that's what happens normally. Now, if you don't have this chloride channel, then a lot of this movement doesn't happen. You don't get the water moving, or very little. Now the mucus is really thick. The cilia stop moving. You're in fact, now bacteria come in. How do you get rid of the bacteria? You can't, right? So the rate of things like pneumonia, all sorts of different infections and things like that is really high in people with cystic fibrosis. So this is the treatment. Now, the gene needed to cure cystic fibrosis is sprayed into the nose and delivered to the lower respiratory tract by adenovirus vector or by using liposomes, which I mentioned earlier. And there's been limited success so far, but eventually we'll figure out probably a way to get some of these genes transferred in. The problem is, like, sometimes you get the transfer, but then it doesn't stick very long. Like the cells may eventually die out, so you have to keep redoing the treatment, that sort of thing. So in vivo gene therapy increasingly relied upon as a part of cancer treatment. So why is this useful for cancer treatment? So about a third of people will in fact get cancer, right? So there's a good chance that, you know, one of us or some of us will get cancer, right? So how do we combat cancer? It's used to make healthy cells more tolerant of chemotherapy. So chemotherapy is really harsh, right? But maybe if you make your healthy cells more tolerant, that's a possibility. You can make your tumor cells more sensitive to chemotherapy. If you can target tumor cells particularly, and there are ways to target, they're rapidly dividing. So there are ways to very specifically sort of target the tumor cells and then introduce genes into them and make them more sensitive. Find a way to introduce the tumor suppressor gene. I mentioned this before, gene P53. Remember, that's sort of a master gene. That's a tumor suppressor gene. When it gets mutated, then you're no longer able to suppress tumors. But what if you could, okay, so that gene is missing in your cells within the tumor, but what if we are able to somehow get that gene back into the tumor cell? Then they should stop dividing, and they should even perhaps commit apoptosis, which is a controlled cell death. All right, so then we're on to the last section here. So this is talking about genomics, proteomics, and bioinformatics. So genetics in the 21st century largely concerns genomics, but there are new technologies and ideas that are coming out now that are combining with genomics. So what is genomics? It's the study of the complete genetic sequence of the human and other organisms. So the very first step is just simply figuring out the sequence of base pairs within the genome, right? So we're doing that. We have machines that can do this at an incredibly rapid rate now, right? If you wanted to know your full sequence, you could pay roughly around $1,000 and have your genome sequenced nowadays, which is incredible. Of the 3.2 billion bases in the human genome, this is an interesting fact. About 98% of your base pairs don't code for anything. They're non-coding. They don't code for protein or anything, right? And you have lots and lots of these repetitive sequences, which I mentioned earlier, like the STRs, right? Short tandem repeats. Second step is mapping the location of genes on the chromosome. So we now know that there are roughly 23,000 genes that code directly for proteins in humans. So I mentioned this earlier, I think last time, that there was this thing called the Human Genome Project. It was sort of, probably didn't get as much press as sending a man to the moon, but it was roughly on that order of scale. It was a worldwide project, actually. It wasn't just the United States, where there was a 13-year effort involved in both university and private laboratories where people were working on trying to essentially decode the human genome, figure out all the sequence of bases, right, within the genome of a human. And it actually came in under budget and ahead of time because the technology got so much better so quickly. So new DNA sequencing technology helped speed the process. This is an understatement. It's just completely sped it up tremendously. New genomes are being sequenced all the time at a much faster rate now. So recently, there's Xenophus labius, which is a very important experimental animal. It's the African clawed frog. People use it for developmental studies to understand how the body develops and things like that. They sequenced this in just 1 year, right? And now this has become sort of old news. Probably it would take you a month or so forth to sequence the whole genome of an organism that you pick. So what do we find when we look at when we sequence the genome? One thing they found was this single nucleotide polymorphism. So polymorphism means that if you look at a particular gene locus, that there are different forms. Poly means many, morph means shape, right? So different forms of genes. And this is very common, right? So between you or I, we may have the same gene, but we might have a 1 nucleotide difference. And sometimes this has no effect, right? That's neutral. Sometimes it actually promotes a protein coding difference. So you might have slightly different proteins. And sometimes someone will just have a completely non-functional protein altogether, right? So this is important. So we're starting to understand this better. May change the susceptibility to disease response to medical treatments between individuals, may raise the possibility. So once we understood that people vary a lot like this, the idea is if you're being treated for a particular disease, a particular cancer or something like that, maybe we should know something about your genes, that particular gene you have. And that may then dictate how you're treated medically, right? So this raises the possibility of designer drugs tailored to individual genotypes. So we might treat one person differently than another person, depending on their genes. Okay, so yeah, so I basically have about 23,000 genes that code for protein. Structural genomics is going to sequence the bases. That's the earliest thing. What we're now on to now is we can do this pretty easily. Well, not easily, but it's doable now. But now we want to know about functional genomics. What do the different what do the different genes code for? So many of the genes are expected to code for proteins, but there are a lot of non-coding DNA, which used to be called junk DNA. There's people don't really call it that much of that anymore because we keep finding functions of the non-coding DNA. Non-coding DNA often codes for genes. First of all, you may not make protein. You're going to make transfer RNA, right? You need genes for that. You need genes for ribosomal RNA. Of course, you need that. But then there are a lot of these non-coding regions that actually are control genes. They control other genes, okay? They may not actually particularly code for a protein, okay? And then there is some stuff that's simply non-coding. So if you look at the human genome, it appears that, well, certainly 98% of the human genome doesn't code for amino acid sequences. But some of this can be, some is transcribed that I mentioned a second ago to ribosomal RNA or tRNA. So obviously those aren't proteins, but they are involved in protein assembly. The rest of the genome consists of a variety of sequences. Some are control sequences, but some of these, again, there's a tremendous amount of these SDRs, these repeated sequences that don't seem to have any function as far as we can tell, but who knows? In the future, maybe we'll find a function for them. I mentioned this, Barbara McClintock. So she looked at these things called, these aren't the same thing as SDRs. These are transposable elements. These are also known as jumping genes or transposons. They can jump around your genome. So about 44% of your genome is actually made of these transposons. Discovered by Barbara McClintock in 1950. They may be driving evolution because they cause rapid mutations and things like that. Then we talked about these repetitive elements, another thing within the genome. You have the same sequence of 2 or more nucleotides repeated. These are the SDRs repeated. They may not be useless. Centromeres, remember the sort of cinched part of a chromosome, that's the centromere. That has a lot of these repeats, these SDRs, and the telomeres at the very end. They're kind of like function the way, you know how if you have shoelaces, you have a little plastic bit that wraps around it so it doesn't become frayed apart. So these repetitive elements are actually, our DNA is linear, so it can start to unravel at the ends, right? So these sort of hold the ends together. And actually, as you get older, the telomeres get shorter and shorter and shorter. And then eventually you start losing genes, okay? Sequences of, and there, of course, there's still a lot unknown here. So, yeah, I think we're pretty much out of time, but at the very end, they talk about redefining the gene. So historically, we thought about genes as simply coding for protein, but it's probably more complicated than that, right? Is all they're really getting at here. There's other possibilities, but we're out of time. So you can take a look at that. And we're also comparing genomes across different animals to help us understand the human genome better. Okay? So take a look at that. It's not really very much to read there. That's just a little bit of the very last section. So we'll stop there. So on Tuesday, we'll have a review for the last exam, which is going to be on Thursday. Okay. And I should warn you, like unless you've already made arrangements to take if you wanted to do an exam a different date, it's very difficult right now because if you go to academic services, they only have a very limited number of slots. Okay.
Can't really do exams on that last Monday.
On
Nobody's gonna be there.
On the Monday, you can't take them?
Well, like the last Monday.
The last
there's, like,
Monday, right.
very, like,
Yeah. So if you want to, I don't know, you can talk to me about it though, okay? If you can't take the exam on Thursday, but it's very, very limited, very, very tough. It's very tough because everyone's trying to take an exam at the very end. All right, thanks, guys.
This looks like,